All right, ladies and gentlemen, we covered a lot of this slide in class for us, or from some of us. But we're going to talk about how alleles are related to the process of meiosis, and how on Earth, if everybody has two alleles for a trait, even parents, how do they pass on only one to their offspring? And the answer, of course, is meiosis, right? It's the formation of gametes. And we're going to visualize that here. But firstly, gametes are obviously reproductive cells, sperm and egg cells. And here we have the process of meiosis. The circle represents a cell, and we're going to have the capital letter T represent the dominant allele, as we do in genetics. And then we'll have the lowercase t represent the recessive allele. So we have in this cell, we have one chromosome that has a capital T on it, one chromosome that has a lowercase t allele present. And then before meiosis, we have to go through interphase. An interphase includes S phase, which is where chromosomes are duplicated. So when that happens, we have two new copies of these alleles. Now, when meiosis 1 happens, we're going to separate these homologous pairs. All right, remember in meiosis 1, it's the separation of homologous pairs that occurs first. So big T goes to one cell, little t to another. And then in meiosis 2, we're going to separate these alleles from one another. Okay, so little t, and big T will be separated. And at the end, we have 4 either sperm or egg cells with only one allele present, and it could either be the dominant or the recessive allele. And during meiosis, the DNA is replicated and then separated into 4 gametes. Remember, the process of meiosis is chromosome number and set reduction. We go from having 2 sets of chromosomes to 1 set of chromosomes and half the number of chromosomes. And in this way, this diagram pictured here on the slide, this is how each parent only passes on one allele, because each gamete, each sperm or egg cell, only has one allele for each trait, as you can see at the end. Okay, so now let's jump into Mendel's principle of segregation. We talked about Mendel's law of dominance and recessive. Now his principle of segregation is kind of like what we just talked about. So Mendel's principle says that every individual carries 2 alleles for each trait. These 2 alleles separate or segregate during the formation of sperm and egg, during meiosis. And that's basically what we just depicted on the last slide in that diagram, and we can see it here, again, depicted the same way. Now, we're already pretty familiar with these terms. We know homozygous and heterozygous thanks to our Amoeba Sisters video last week. So because we have 2 alleles for each trait, we can either be homozygous for that trait or we can be heterozygous for that trait, depending on the combination of alleles we get from our parents. So homozygous, homo, the prefix means same. So homozygous means that the 2 alleles are the same, either 2 dominants or 2 recessive alleles. And heterozygous, the hetero prefix here means different. So you have 2 different alleles for a given trait, and there are only 2 different types, dominant or recessive, so it's got to be only this genotype. Speaking of genotypes, we know about genotypes and phenotypes thanks to the Amoeba Sisters from last week. A genotype is basically the genetic makeup of an organism, right? The combination of alleles that organism has in its genome. A phenotype, on the other hand, is the physical characteristics that those genes give off, those alleles express, right? Like having blue eyes, having dark skin, having blonde hair. These are all phenotypes, being very tall. Okay. Now, the phenotype is dependent on the genotype, right? We are only looking certain ways because we have the genes or the alleles to look a certain way. In my example here, we take it back to Mendel's pea plants. This pea plant, or both of these pea plants, have the allele for being tall, and because the tall allele is dominant, they are both tall, despite one being homozygous and the other being heterozygous. So if we look, we have the genotypes, capital T is equivalent to the phenotype tall, and capital T, lowercase t, is also equivalent to the phenotype tall. So if we just looked at the phenotype, we couldn't tell a difference between these two plants. But if we look at the genotypes, we can see that one is a hybrid and one is a purebred. And lastly, little t equals dwarf, right? 2 recessive traits is what you need to express the recessive phenotype. 2 recessive alleles, I'm sorry. So, if we know the genetic makeup of the parents, can we always predict what they might produce as offspring? Or even better yet, can we predict the probability that they'll have a particular offspring? And probability plays a big part in genetics, right? Probability is just the likelihood that a particular event is going to occur. Now, let's talk about how probability works with Punnett squares. We got a little taste of this from the Amoeba Sisters video. So we know how Punnett squares work. Punnett squares is a diagram that shows allele combinations that might result from a genetic cross. The alleles for the first parent, as we know, go across the top of the square. And the alleles for the second parent, of course, go down the left-hand side of the square, as we did last week. And then all the possible gene combinations, or the allele combinations, we represent with letters that are alleles in the middle. Capital represents dominant, lowercase represents recessive, and then we combine them. Okay, and we see that there's a 50-50 distribution here, right? So we have a 1/2 chance of getting a homozygous dominant offspring, or a 1/2 chance of getting a heterozygote offspring. Okay, now we're going to stop here, and I'm going to take us out of the slides. So in your study guide, what I'd like you to do, okay, if you are up here at the top of your study guide or wherever you are, and your study guide looks like that, I want you to click these 3 lines over here, and then I want you to click the principle of independent assortment, because that's where we're going to go next. Okay, we're going to go to this section of the study guide. So once you're there, that's where we're going to start up again in the next screencast. Now, what I want to make you aware of is this slide, 25, 26, 27, 28, 29, and even 30. These are all genetics problems that you are to do on your own for studying purposes and for the test. So please do those on your study guide. Catch you guys in the next one.